You're listening to Medscape's In Discussion series on endometrial cancer, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Ursula Matulonis, Chief of the Division of Gynecologic Oncology at the Dana-Farber Cancer Institute and Professor of Medicine at Harvard Medical School. She is the first recipient of the Brock Wilson Family Chair at the Dana-Farber Cancer Institute. She co-leads the Gynecologic Cancer Program within the Dana-Farber Harvard Cancer Center, the Ovarian Cancer Specialized Program in Research Excellence, SPORE grant, from the National Cancer Institute within the Dana-Farber Harvard Cancer Center, and an NCI P01 program project grant that is focused on targeting replication stress and high-grade endometrial cancers. Relevant disclosures can be found with the episode show notes on Medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hi there. I'm Dr. Ursula Matulonis from the Dana-Farber Cancer Institute in Boston, Massachusetts, and welcome to Season 1 of the Medscape In Discussion Endometrial Cancer podcast series. Today, we're going to discuss antibody-drug conjugates, or ADCs as they're known, and their use in endometrial cancer. And it is my extreme pleasure and honor to introduce my guest, Dr. Kathleen Moore. Dr. Moore is the Deputy Director of the Stevenson Cancer Center, and she is a professor of gynecologic oncology at the University of Oklahoma College of Medicine, a true pioneer in our field. It's really an honor, Katie, to have you here, and welcome to the Medscape In Discussion Endometrial Cancer podcast.
Thank you for having me. I'm happy to be here.
You know, the use of antibody-drug conjugates in endometrial cancer is at its beginning stage in terms of the clinical use, but the clinical use has already started. And it really is a really exciting field, and you've done a lot of work on this. And really what I want to talk about in our podcast today is to focus on the clinical aspects of the drugs that our oncologists can use to treat our patients with endometrial cancer, specifically trastuzumab deruxtecan, which has now appeared on the NCCN guidelines, and kind of compare and contrast a little bit to a drug you know well, I know well, mirvetuximab soravtansine, the first antibody-drug conjugate to be used and to be used for ovarian cancer. And obviously, we have antibody-drug conjugates to use in cervical cancer, but the field is really just starting to open up in endometrial cancer. Today, I'd like to cover trastuzumab deruxtecan. And Katie, I know you've worked on other promising HER2 antibody-drug conjugates, to have our audience think about potential mechanisms of resistance and how we can think about them, potentially overcome them. Think about other promising targets for endometrial cancer, FOLR1, TROP2, B7H4, CDH6, etc. Think about some exciting antibody-drug conjugate combinations, and then obviously anything else you'd like to mention. So let's get back to trastuzumab deruxtecan, and can you tell us about the drug and how you're using it in your practice?
Sure. I think we're just starting to really figure out how to use trastuzumab deruxtecan in clinical practice. As you alluded to, it's now in the NCCN guidelines based on the data that was presented by Dr. Funda Merrick-Berstrom at ASCO and then ESMO in 2023, which is exciting. But, most oncologists have never used this medication because it's really been in breast cancer and gastric cancer. And so it's a really nice opportunity to talk about it in a little bit more detail as it's now available for us to use. So it's an antibody-drug conjugate, as you alluded to, and it targets HER2. And we've actually known about HER2 or ERBB2 as a target in endometrial cancer for quite some time, for actually over a decade. And my colleagues, Michael Bookman, Alessandro Santin, and others have been working on HER2 targeting, mainly in serous cancers, for a lot of their careers. And it's been a little bit of an elusive target till antibody-drug conjugates. With just monoclonal antibodies such as trastuzumab and pertuzumab, the response rates were very low. And also, really finding tumors that were what we traditionally called HER2 high, which was, it's evolved over time. That's a whole nother podcast, but was HER2 2+-ish positive or 3+, kind of limited the population. But even with that, we have ongoing studies, one ongoing right now by Britt Erickson, that's Energy GY026, that brings in pertuzumab and trastuzumab with chemotherapy and to follow in serous tumors that are 2+-ish positive or 3+. And that's an important study that should finish. But it's going to be a little hard now that we have ADCs. And so ADCs have roared on to the scene with the Destiny Pan-Tumor study, which, just to remind the audience, was a basket trial of trastuzumab deruxtecan in 2+ or 3+ by gastric scoring, which accounts for the more heterogeneous HER2 expression in non-breast cancers. That's why you use that, and that's why they used it here. Then it looked at ovarian, endometrial, and cervical cancer. And just also to remind the audience, trastuzumab deruxtecan targets HER2, and it has a deruxtecan conjugate, which is a topoisomerase 1 inhibitor, which isn't a chemo we typically have used in endometrial cancer, and has a drug-antibody ratio of 8, which means there's 8 molecules of chemo on each antibody-drug conjugate. And so when they used this in endometrial cancer, in the 40 patients who were reported, the overall response rate was like 58% in the recurrent settings. These were patients who had at least seen one line of chemotherapy. They had to be second line and beyond. Didn't really characterize their prior therapies other than that, but they were at least second line and beyond, which I think, as we all know, 58% response rate is pretty much outstanding, which is why it's NCCN listed. So you came on based on local testing, gastric 2+, 3+. Then they did central confirmation where they did see drift. And in that 13 of the 40 patients who were confirmed 3+, the response rate was 85%. And in the 17 patients who were HER2 2+ confirmed, it was 47.1%, both of which are still really good. And then, of course, was reported across the other gynecologic cancers as well, which we can maybe get to. But endometrial was certainly the star. And we're going to see this come into bigger phase 3 trials in endometrial cancer in the front line coming soon. So, I think that's where you're going to see a very rapid shift in how we use this and other antibody-drug conjugates with these trials. But for now, I think people are using it in the recurrent setting, as is the NCCN listing.
And I think in addition to those pretty remarkable response rates, also having a median progression-free survival that was not reached, in addition to a median duration of response. And then, there's the status trial from Japan that also looked at trastuzumab deruxtecan, but in uterine carcinosarcoma, also showing pretty remarkable response rates. And in that trial, the investigators even went down to HER2 low, 1+, but in the NCCN guidelines, it's 2+ or 3+ HER2 via immunohistochemistry. And I think that's another important point for our audience, is that this is not by FISH. This is not based upon HER2 mutations, but it's based solely on immunohistochemistry, which I think our pathologists are not necessarily always doing. And you also pointed out the different types of ways of scoring, gastric versus breast, but then there are different antibodies out there as well. Do you have any recommendations, or do we just say, okay, pathologist, please check HER2 IHC? Any recommendations for pathologists in this setting?
So that's a great question. And I think it's still one of the unknowns, really, for us with endometrial cancer, where we're catching up, but we're really behind breast cancer and gastric cancer and others who've been really looking at this and correlating it with outcomes for quite some time. We, of course, have fairly large samples looking just for those true positives, again, the 2+-ish or 3+ when we're talking about monoclonal antibodies. But antibody-drug conjugates, we have to kind of start over, which is why I'm glad we have the NCCN listing, because really the patients who have HER2-driven tumors, I think we feel that they're worse prognostically, and we know that endometrial cancer is on the rise, especially among patients of color, and many of those patients don't have access to trials. And so I'm very supportive of this being made available so that the patients can have access to what really can be a transformational drug for them. But I'm also equally passionate about us continuing to do clinical trials, even in the recurrent setting, because we really don't know the answer to the question that you just asked me. I'm involved, in all full disclosure, in a competitor, which is BioNTech's HER2 ADC BNT323. And in that study, we're allowing 1+ and up, and even amplified, really anybody that has HER2, we're allowing to come on, and then they're doing central reevaluation through a number of methods to try and figure out, is there anyone that shouldn't have access to this? And I don't think we know the answer to that yet. Certainly the updated data that Dr. Bertson presented at ESMO, where she did break out the 2+ versus 3+, does look different. So it does look like there's a difference there, which isn't that surprising. But the numbers are very small, and we haven't really done kind of our normal study in a homogeneous population. Here we would expect patients to maybe have been exposed to immunotherapy, real homogeneous population, and what's your response rate, and then break it down by the different IHCs. So I don't think we know yet enough for me to say, hey, pathologists, do this assay and this methodology, other than I would say that HER2 IHC should be a part of the standard of care panel for any patient with, actually at this point, any gynecologic cancer. I would say we're catching all ours up, but certainly endometrial, you want to know your mismatch repair proteins, and you want to know right now your HER2 status, so you don't miss any of these patients and their availability to get trastuzumab deruxtecan on compendium. We can't say on label at this point, or access to a clinical trial that allows 1+ and up. So I really feel like we have more work to do to figure out who benefits from monotherapy. And then just like, and you know this as a medical oncologist, but in breast cancer, there's so many interesting combinations coming out with these deruxtecan payload ADCs, kind of sequencing in scientifically driven ways, a combination therapy for those HER2 low tumors. So we need to pay attention to that as well. So I feel like we still have a lot to learn.
And I think, just before we go on to kind of thinking about combinations and ways of overcoming, resistance mechanisms, just a few more points on trastuzumab deruxtecan, just because it's being used in clinical practice. I agree. If the NCCN guidelines based upon 40 patients, now we also have the status trial in the in uterine carcinosarcomas, which adds another few. But important things to check, right, are baseline chest, abdomen, pelvic CT scans, checking an echocardiogram because it's a HER2-directed therapy, so there can be cardiac toxicities. And then once patients start, we don't have an FDA approval here. Like you said, it's on the NCCN guidelines. And even in the FDA package insert, it's a little bit not clear about how often patients should be monitored, but certainly based upon the PanTumor02 study, patients did undergo restaging, which included a chest CAT scan every 2 cycles, and they had an echocardiogram every 4 cycles. So just before they started the 5th cycle. Side effects, certainly have to watch out for diarrhea, neutropenia. There can be hair loss with this drug. And then, around a 10%, maybe a little bit higher risk of interstitial lung disease or ILD, which can be pretty significant in this agent. What's clear on the FDA guidelines, if you see any kind of interstitial lung changes and it's a grade 1, patient's asymptomatic, that patient should not receive more trastuzumab deruxtecan. You have to watch them until the chest CAT scan clears. And if they're grade 2 or higher, you have to stop the drug, which is so different than other antibody-drug conjugates that we use in other gyn cancers, especially mirvetuximab, where grade 1, you can watch and continue to treat. Any thoughts about that?
I'm so glad you brought that up. I'm a product of Joan Walker, who always was like, I'm going to tell you my mistakes so you don't make them. But this is a place where I did make a mistake, tragically. So mirvetuximab, we see ground glass opacities or patchy infiltrates or whatever, and patients like, I feel great. And they don't have any symptoms, and we just kept treating. We just didn't do anything. And in some parts of the country, you just see these called on chest CTs all the time. Inflammatory changes, non-specific, could be inflammatory or infectious, clinically correlate. All these things that show up on the chest CT that, I would be quite honest, here we were like, oh, not cancer, patient feels great, we're going to keep treating. We were not looking to hold asymptomatic, normal O2 sat, some inflammatory change on the CT scan. And with this class of drugs, with deruxtecan payloads, it almost, what I would just suggest is you just make it a checklist on your CT, like no cancer, yay. Patient feels great, yay. Any changes on the CT that they're calling inflammatory or fibrotic or questionable, clinically correlate, just make yourself a checklist. And if that's yes, you hold, and you need to call the pulmonologist. It's not a referral because they will not see these patients for like a month because they're fine. It's a phone call like, hey, this is what's going on. I'm holding the patient until you either look at the CT or see the patient and tell me it is or isn't pneumonitis. And that's uncomfortable because these patients have this disease that really kills them, and you don't want to hold something that's working. But I'll tell you, when you hold in this drug that's working, they don't lose control while you're waiting to get this worked up. They actually do fine. So you actually have grace to work it up and make sure you're not treating through grade 1 pneumonitis where the patient does not want to stop. They feel fine and they are baffled why you are worried about them. But you have to make this a checkpoint for yourself, and then it's safe to use. But if you don't do that, get into trouble, and that's what happened to me and my patient, and I don't want it to happen to any of you. And there's no need. We learned this from breast, so we just need to communicate this so we don't have to have the same learning curve that breast cancer had. But it's tricky. It doesn't say pneumonitis on the chest CT. They don't go, hey, alert. They don't say that.
If you had the opportunity to take a look at the chest images just to see what they're calling, how they're calling it, that's really important as well. And I think you're right, calling out the pulmonologists who are super busy. But, even the FDA package insert, which is not directed to us in GYN cancer. I mean, that's part of the issue, is that we just need to educate ourselves about the toxicities of these drugs. And sure, they're very, very impressive, based upon PAN-Tumor02 and the status trial for endometrial cancer, but they do have pretty serious toxicity. So we just need to keep educating ourselves. So that, to me, Katie, moves on to all the different HER2-directed antibody-drug conjugates that are being developed right now. And I think there's always this conversation, well, you've got trastuzumab deruxtecan, great activity. Why study other HER2-directed antibody-drug conjugates? And I think, certainly one reason would be to avoid and lessen the toxicity profile that we see with this drug. Can you talk about maybe some of the drugs that you're testing, that you're excited about, and that you see as promising next antibody-drug conjugates to HER2?
Well, there's a lot, and you make a great point. And I think this is sort of an issue across ADCs, and Dr. Anthony Tolcher gave a great talk on this at ASCO Breakthroughs last year, where he talked about the ADC bubble, that there's 20 TROP2 ADCs in development and 10 folate and however many HER2. I haven't even counted them all yet. And the question is why? If they're just Me Too agents, of course there's competition, so you need a few. But, do you need 6 Me Too agents? And the answer is no. And then for a lot of reasons, it's just unnecessary. It diverts investment, in small biotech. If everyone's developing a Me Too, and you already have 4 ahead of you, then let's move investments in novel agents. I think this is sort of beyond the top of this podcast, but we need to stop all these Me Too drugs and then focus on things that are really differentiated, because you may have the same target, but it's a really differentiated molecule. And then the differentiated molecule needs to work better. And probably some of the best examples of that are in folate, which we can talk about in a minute. But HER2, I think, is coming. We've kind of already seen the evolution of HER2, though. We started with trastuzumab and Tansy, which had a microtubule toxin payload and was an anti-microtubule. And the DAR was 3.5, I think, to 1. And that's been tested through several basket studies in endometrial cancer with pretty, I wouldn't say negative, but, the best response rate we've seen with that agent for endometrial cancer was presented last year at ASCO and was a response rate of 21%. Now, I will say that study did some really beautiful translational or correlative work with off-treatment biopsies. It's really exciting, which we could talk about. But 21% is, not that great. But then you change the payload, you change the DAR, the linkers are different, and you have a differentiated molecule like trastuzumab deruxtecan or BNT323, and you get into these response rates north of 50%. So I think that's quite important. So those are probably the 2 leading ADCs that I think are in the space kind of right now, both of which have registration-enabling studies in both frontline and recurrent settings. Not everything's in public domain, so I can't be overly specific. But they are different in their biomarker selection, and they both have pretty robust plans for looking at the biomarker. And so I think both of these are quite important to get rolling out globally so we can make sure that we're testing broadly, just like we did with folate receptor alpha. We know what the best test is, if it matters. Maybe it doesn't matter, and you just need to know if someone's HER2 something. And then what's the best treatment line to use these drugs in? So I think that's kind of where we are now with HER2. There are some interesting bispecifics coming behind this that I'm interested in that are still early, and potentially some more differentiated molecules coming up in the pipeline. But in terms of closest to clinical action, I think are those 2.
Yeah. And you've done so much work on this. And we've already talked about FOL1 just a little bit. It's important, certainly in ovarian cancer, but there are multiple drugs and ADCs that are being tested right now for folate receptor alpha and then TROP2. Any other targets that interest you, and any of these targets particularly of interest to you?
The next nearest thing are the TROP2. There are 2 studies with 2 different TROP2 ADCs, and they are different in terms of their payload, are about to launch in the recurrent setting. So these are different drugs. Even though both target TROP2, they have different payloads. One's an SN3 payload and one is a topoisomerase 1 payload. So similar but not the same. And I think their side effect profiles and perhaps efficacy could be different. So I think TROP2 and endometrial is probably the next nearest thing to come forward apart from HER2. And then closely behind are the folates. Now, mirvetuximab, which you and I both know and love, so we have to say that out loud, conflict of interest. Love Mirv.
Yes, we do.
Has had some experience in endometrial a long time ago. And finding those FR alpha high was mainly in serous. It was pretty infrequent, and we didn't really see a lot of responses. I think that's just the truth. So that's probably less interest in that particular agent, but not in folate. When we think about other folate agents, there's a lot, as I said, kind of Me Too agents out there, but probably from my standpoint, 2 of the most differentiated agents are the new immunogen 151, which is a biparatopic antibody. So it hopefully binds lower levels of folate better, and it has an updated payload. And so that's up and coming in phase 1 and is enrolling in endometrial and ovary. And so I think we'll get a sense from that, whether or not that has a play in endometrial. And neck with that is Profound Bio's PRO1184, which targets folate, but has an exatecan payload. Also has enrolled endometrial and ovary, and is probably about the same place as 151 in terms of dose escalation and expansions. So I haven't seen any data. I don't have any insight or information on how they're looking in endometrial, but I'm just excited about them because they are really differentiated molecules. And so, they are really the next gen. And there's a few others out there that are a little earlier, but I'm excited to see some of the early presentations of this. And I hope we'll see some of those this year. I'm very excited about B7H4. I don't know if it's going to be an ovary or endometrial focus yet, but I'm excited to see some of those read out. Those are also about the same place as the 2 drugs I just mentioned, maybe a little further along. So we should start to see some readouts of those expansion cohorts. I think those are our top ones in endometrial: HER2, TROP2, folate, and B7H4. CDH6 probably less so in endometrial, although I'm not ruling it out. May not be as big of a play.
Yeah, tons of work to do. And obviously, I know we just have a few minutes left, but in terms of the importance of looking at all these medications as also connected with the different mechanisms of resistance that patients' cancers have to antibody-drug conjugates, in terms of the measurement of the antigen expression and whether or not you need high levels or low levels, the tumor heterogeneity, which certainly you see, and especially certainly see in a cancer like endometrial carcinosarcoma, and then the changes over time that occur with antigen expression, and whether or not the ADC actually engages with that antigen. So there's just lots of work to do. Payloads, right? We mentioned a lot of different payloads, and thinking about sequencing. I'm going to wrap up by asking you, Katie, favorites in terms of antibody-drug conjugate combinations?
We've looked at a lot of combinations with the ADCs, and thus far, mainly in ovary, they've been additive. We haven't really seen synergy with ADCs to date, maybe with bevacizumab and mirvetuximab with 2. That may be one place where I take that back. We have seen some synergy. But with this new class of ADCs with topoisomerase 1 payloads, there is just quite a bit of preclinical and early clinical rationale for combinations and sequencing a topo 1 and then a topo 2 inhibitor, like using temporal sequencing, and really thinking about synergistic targeted therapies with what is a chemo. It's just targeted chemo. So I do think that this is a place where we may see DNA damage response inhibitors come back, whether it's PARP or some of the newer agents that are coming down the pike, USP1 inhibitors, Pol Theta. I'm working on a couple of super novel drugs that preclinically look like there's quite a bit of synergy with some of these topoisomerase 1 inhibitor ADCs. I think that we're going to see some of those come back around, and they'll be particularly important, not for everyone, but maybe HER2 low does need a combination. So I think we're going to see some of those combinations. So I would say my favorite thus far are these DNA damage response inhibitors, but I constantly am learning actually from some of your partners. I learn from Joyce and Panos every day. And your DNA Damage Response Institute has led a lot of this work. So whatever you're working on has been my favorite thing, actually. But I think it's not just the combos, it's figuring out the patients who need a combo and then not using combos in the patients that don't need a combo. And I think we're going to see better discernment of that moving forward. So I'm pretty excited about those combinations coming. I just think it's a really exciting time across gynecologic cancers, but specifically in endometrial, where for so many years, we really just were not making any progress. It was just paclitaxel and carboplatin, and maybe you'd add a little bevacizumab if you could get it authorized on NCCN. But really, we're just sort of stuck. And then all of a sudden, we've gotten unstuck in a very rapid way. So immunotherapy with understanding subgroups, even beyond the TCGA. How long have we been talking about the TCGA? We're finally acting on it and then realizing it's even way more than 4 groups. The non-specific mutation profile, that copy number low MSS group, is probably 4 groups. And we're designing studies for those molecular subtypes that are actually working. And the HER2 ADCs and these other ADCs are bringing agents in that actually work and work really well. We have to pay attention to the toxicity. So I didn't mean to scare people, but I want you to pay attention to it, because otherwise these drugs are transformational for people that otherwise would not be alive.
True. Well, it's been a great discussion, and thank you so much, Katie. It's been a real pleasure. It's always a real pleasure talking with you. And so today we've talked to Dr. Kathleen more about different aspects of antibody-drug conjugates in endometrial cancer. We talked about some of the medications that are available to us right now, such as trastuzumab deruxtecan through the NCCN guidelines, other promising HER2 ADCs, specifically that Katie's been working on, other targets for antibody-drug conjugates, promising combinations, and it's been a great discussion. So please take a moment to download the Medscape app to listen and subscribe to this podcast series on endometrial cancer. This is Dr. Ursula Matulonis for the Medscape In Discussion Endometrial Cancer podcast. Thank you so much.
Thanks for listening to Medscape's In Discussion Endometrial Cancer podcast series with our host, Dr. Ursula Matulonis. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out Medscape.com or the Medscape app for show notes, links, and more information on endometrial cancer.